Okay, well, we're getting set up. I'll introduce our next speaker. Our next speaker is Dr. Jeff Pettis. He's a former research leader at USDA's Bee Research Laboratory and the principal of Pettis and Associates and the president of the Scientific Commission on Bee Health of the International Federation of Beekeepers Associations. Jeff is a groundbreaking researcher on the behavior and chemical ecology of bees. His research has led to a greater understanding of colony collapse disorder in honeybees and the role played by neonicotinoid insecticides.
Thank you. Good morning. It's a pleasure to be here. I'd like to thank Jay and Beyond Pesticides for the chance to come and speak to you. I'm an entomologist by trade, and I studied almost exclusively honeybees, but in the role of honeybee health, we began to recognize the role of pesticides in bee health. So I've served on both European Food Safety Administration panels evaluating the health of bees, and also, as the previous speaker talked about, Science Advisory Panel to EPA on the impact of pesticides in bees. And I'd like to take at the beginning just a moment to say something about those Science Advisory Panels. So we made two strong recommendations to them. One is that they only used honeybees as their model, adult honeybees. So adult honeybees were the model for impact on pollinators. So we recommended that they study immature honeybees or immature bees in general. Why would that be? Well, same in human health. We see more effects on immatures than we do on adults. The second recommendation was that we use other bees other than honeybees. So, and I'll get to some of that in my talk. So look at immature bees and also use something other than just honeybees in predicting whether there was damage caused by pesticides. So I've got a case study from Queens. I had the opportunity to work on honeybees in New York City, and you'll get a little bit of that flavor in a minute. So this is an overview of my talk. Pollinator declines in general, I'll be very brief about that. The challenges to pesticide risk assessment, a bit about EPA and how they make decisions, and then I'll contrast the biology of honeybees, bumblebees, and solitary bees, because they're quite different. Then I'll get to my study in New York City parks, which has a local flavor, and then some conclusions. So colony collapse disorder in honeybees began in about 2007, and I was heavily involved in that. The major outcome of that was grabbing the public's attention, and pollinators became kind of a symbol of what was happening in our environment. Colony collapse disorder in particular is only in honeybee colonies. It doesn't affect wild other bees, but it certainly served as a wake-up call. And what I think is happening in general with pollinators is they need food, they go to plants to pollinate, so you've got the pollinator, you've got the plants that they depend on for nutrition, and in many cases you've got pesticide exposure going on in those plants. So it's this three-way interaction that I've been most involved in. But there's two parts of this three-way puzzle that are missing, and in looking at impact on pollinators, we can't ignore the fact that habitat destruction, just changing the environment, farming, urbanization, habitat destruction has a major impact on especially native pollinators, pollinators that can't move. Up to 70% of native pollinators nest in the ground, and when we disturb that habitat, that has a major impact. And also pests and diseases are not in that three-way interaction. So pests and diseases obviously impact bees. I'll just direct you to this study. It has some major policy recommendations globally for what we can do to improve pollinator health. I had the opportunity to serve on this as a United Nations panel publication, and just type IPBES, an Intergovernmental Panel on Biodiversity and Ecosystem Services. Just type in IPBES pollinators, and you'll get this report. It gives you some tools to take action. So, a little bit about the challenges of risk assessment and what EPA is trying to do in risk assessment. They have a mandate to protect all pollinators. So that one of their goals is pollinator protection. The other question is, are honeybees, in fact, the best model? And I already know the answer. You know the answer. Honeybees are good, but they're not the best model. And how do we use multiple lines of evidence to look at what's happening with pollinators? This just shows you some of the diversity that we have in bees in general. Honeybees are pictured on your left. Ground nesting bees that I've already talked about represent about 70% of the native bees in the US or North America nest in the ground. We do very little work on their exposure to pesticides. You may be familiar with alfalfa leafcutter bees. They're used commercially to pollinate alfalfa. Stingless bees occur a little bit in southern US, but mostly in the tropics. And the solitary bee osmia is a bee that you may be familiar with. It's the one that garden centers sell little houses where bees nest in tubes. This is osmia or blue orchard bee. But all these have distinct life cycles. And I'll just use these three to highlight that. So honeybees obviously have large colonies. There's a single queen that is the head of up to 30,000 individuals in an individual beehive. Bumblebees are also social. They have smaller colonies, 300 individuals. And solitary bees, it's usually one female founding a nest, and she's producing all those offspring within the nest. So that's a solitary bee. But all three have something else in common, and that is that they all use pollen from the plant as their food source, their main food source, that and nectar. Some of the differences are in the flight range, the size of the colony and the flight range. And so bumblebees and solitary bees tend to fly shorter range, whereas honeybees are generalists and fly much longer range. And this comes into trying to understand the exposure of bees have to different pesticides. In the bottom, that bottom picture, that's a solitary bee like an osmia that's gone out, collected pollen, packed it into a little pollen ball, and you see an egg that's attached to each pollen ball. That egg will hatch, feed on that pollen, and develop into a male or female bee. That's the route of exposure for many of these pesticides, through the pollen or nectar. So EPA, we heard a bit about risk assessment and risk management. I've dealt a lot with the risk assessors in EPA. They're the ones that simply try to answer the question, is the product toxic or not? So the risk assessor, the risk assessment makes that one determination. What's the toxicity level of that product? And then the risk manager takes into account all these other factors, not just the toxicity of the product, but whether it has economic benefit and public values and all these other things. And it's actually the risk manager that we need to have most of the influence on. Because risk managers weigh all this other evidence and then come to some conclusion. And it's often not based on what the risk assessment says. The risk assessment may say this product is toxic or somewhat toxic, but the risk manager says, no, but we have other evidence, the farmers need it, and da-da-da, and they'll make a risk management decision which says, no, we can use the product. The other thing I want to say about EPA's risk assessment process and risk management process is we only test one compound at a time. We ask the registrant, Bayer, or whoever, to test one compound at a time, and guess what they test? They test the active ingredient. As was pointed out in some of these other talks, they don't test the formulated product. The Europeans are at least ahead of us in that sense that they do require that the registrant test the formulated product, and those adjuvants that are in much of the formulated product can have effects. So we need to ask EPA to do a better job of testing the formulated product. Okay, so now I'll get into this study where we were looking at a question of using imidacloprid, which is a neonic, and how it might move through red maple trees in New York City. They were using this product to try to control Asian longhorn beetle. An Asian longhorn beetle is invasive from China, and the action by APHIS, Animal Plant Health Inspection Service, was to cut down all the infected trees. Well, once you cut down all the infected trees, there was trees nearby that may need protection, and their way was to use pesticides to treat those nearby trees and try to save them, or try to save them as part of this control program. In very brief, You don't need to understand the dynamics other than the fact that the damage is done inside the hardwood tree, so that the female lays eggs in the branches, it burrows inside the tree, and by using a systemic in the tree, you could potentially kill the insect, the Asian longhorn beetle, and control it. So we worked in Staten Island and Queens. It was a 3-year project, and we collected samples of leaves and pollen and things like that, and tried to determine the proportion of the summer bee diet that came from red maple to see whether there was adequate exposure to bees that would be problematic. So this is a picture of the New York City area. We're up along Central Park now, they're in Manhattan, and over in Queens we have blue dots and yellow dots. Those are 1-year and 3-year sites, and the same on Staten Island. We have 1-year sites and 3-year sites. The 3 green dots are our control sites outside the treatment area. They treated the trees for 3 years in a row, either by soil injection or by injection into the trunk. So within a 3-year time span, because we started with 1-year and 3-year trees, we could follow a 6-year cycle. So we had an advantage there. We only ran the study 3 years. We're starting with 1-year-old things and 3-year-old trees. We were able to cover a 6-year time span. Now, if you're going to work in New York City, what would you need to put bees in New York City? This is in Queens. Of course, you're going to need some forms. Well, we got permission from the cemetery owners in Queens to put the bees there. But when we asked the New York City Parks, we worked in New York City Parks as well, and we asked the New York City Parks, what do we need to put live bees in these parks? They said, well, of course, you need a live animal permit, animal exhibit request. And so they said, are you part of a rodeo? Are you part of a parade? And we're like, no, not really. We're here to do research. So we had a lot of fun filling out the forms here on the Group W bench, and we got the form almost filled out, and we were very happy with our filling out all the blanks, and we came to one question that kind of stumped us, and they asked us, well, what are you going to do with the bee poop? What's your plan for cleaning up the bee poop? And this was the question. Describe methods of sanitary control and waste removal. Attach additional sheets if necessary. Think about elephants on parade or horses and stuff. So we had to have a plan. We had to have a plan for what were these 30,000 bees, and we put 20-something colonies in New York. What were we going to do to clean up behind our bees? And we convinced them we didn't need any cleanup. But so we got the permits. We did the study. It was very interesting, just in general. We did take the public safety aspect of this fairly seriously because we put these hives in very remote areas and tried to hide them. But anyway, so we got permission. We got our permits. We put them in different cemeteries and in city parks. We're going to use a small colony of honeybees. On the front, you see a pollen trap. It's a trap when the bees walk through. It collects some of the pollen incoming into that colony. We're going to use that to look for pesticide exposure. I also want to point out something in this picture. There's not a single tree in that cemetery. This is I'm not sure if it's Beth Israel, I can't remember the name, but this cemetery had been completely, they'd taken down every hardwood tree in that cemetery as part of the control program. And so we were trying to look at ways that they could maybe prevent taking down the trees. Our target tree was red maple, and red maple is interesting in that it produces female flowers seen at the top and male flowers. And usually you have male and female trees that are separate, but sometimes you can have both genders on the same tree. And for those of you on yesterday's, we had the field day. When we moved to the bus and they had that forest part, all those trees were red maple, and they were just coming into bloom. So they were red. They had red buds. That was red maple. I'm taking a little aside here. If you become a beekeeper, one thing you begin to notice, really connects you to nature because you begin to notice everything that's flowering and when it's flowering. And anyway, we got into this study because red maple is a very early spring plant, and it's really important for bees and other pollinators. So we collected a number of samples. 3 years of treatment of the trees, either soil or trunk injected. And then we had the opportunity, so we get 2 years post-treatment by following these plots. We collected leaves, whole flowers, and of course, the pollen that the bees were collecting. It's just a study. Jody, pictured on the truck, was a graduate student at the time, Jody Johnson, and Dr. Robin Rose helped fund the study. She's from APHIS, pictured there on the ground. We did determine that in the springtime in this area, up to 50% of the honeybee's diet and other pollinators, I would assume, came from red maple pollen. So it was a dominant plant, and it composed a lot of the diet. So if you're going to look at risk assessment, obviously this is an important plant. I'll show you just a bit of data. The speaker last night said, you know, they don't like to use PowerPoint. As a scientist, we have to show a little bit of data. But it's limited, it's very limited, believe me. So this is from the leaves, and along the bottom you have 1 year, 2 year, 3 year, up to 6 year. And the green is trunk injected levels, and the darker color is soil injected. And it's very obvious that trunk injected, in the leaves at least, you get a lot of imidacloprid, and it begins to decline over time, which is a good thing. In other words, by 6 years, 3 years post-study, the level in leaves had gone down. So it did, and in soil, though it was slower acting, it came up and peaked in year 4. And so there is a difference between soil and trunk. But the thing we're really interested in is what are the levels in pollen. And if you look, the blue bars are on 3-year soil and 1-year soil, and they're reversed there. 1 year should come first, but there's only detectable levels in the soil-injected trees. So if we move towards trunk injection, we're going to have much less impact on pollinators. So that was one of the major findings of the study. So there is a way to use this particular product in this particular setting and reduce the exposure. So again, the question we had was, what is the level of exposure? Trunk was much better. There was some degradation over time. And could we save these trees? So that, to me, is the policy question of this particular study, is what is the value of these mature trees in a cityscape? Is it worth this bit of pesticide use in maybe a safe manner to save these trees? And so those are some of the questions I throw out as a policy, and I don't have answers for them. APHIS did use this information, and they continue to try to control Asian longhorn beetle, and I think we gave them tools to maybe use this product in a safer manner. So just some concluding ideas. So what's wrong with this field? We don't have time to go into what all is wrong with this field. Our agricultural system is a bit out of whack, and we know that. This is Roundup Ready soybeans, but I think we can begin to take control of this system. It needs to change. It has to diversify. We know that. But there's one thing as consumers we can do to begin to change this dramatically, and that is just consumer choice. Yep. We ourselves can stop insisting on perfection in the foods that we eat. So that's a major thing that we can do as consumers. I'll say the next thing that I think is wrong with the agricultural system is that the farmer is chasing that last little bit of yield, which is not really necessary. I mean, they're so overburdened with debt and stuff that they're chasing this last little bit of yield that's not really necessary. And certainly organic offers alternatives to that. Not very sexy, but we've lost a separate extension service that goes out and talks to farmers and gives them advice. All the advice comes from Bayer and Monsanto, so we lost that. And then the last thing is greed, and we know that. So these are the things, but we can make a difference in that first one dramatically. Okay, just summarizing, we have to remember that all pollinators are threatened. It's a global issue. Honeybees may not be the best model. We need to test multiple products and also the formulated product if we're going to look for safety or just reduce use in general. Neonics, I brought you an example of neonic use in mature urban trees. Is it justified? It's a good question. I don't know. I tend to like the trees, so I would think that maybe we can use that safely to save some urban mature trees. And lastly, I'd say that we must reduce the use overall of pesticides, both in human health and in pollinator health. So thank you very much.
Okay, while we're getting set up for the next talk, I want to remind you that both of these speakers will be speaking in the workshop sessions, and that you'll be able to ask them questions then. Okay, our next speaker is Dr. Michael Hansen. He's an evolutionary biologist and senior scientist at Consumer Reports Consumers Union. Michael is credited by friends and critics alike as being the brains behind Consumer Reports' campaign on agricultural biotechnology, and all of us appreciate his guidance. Thank you.
Thank you. Actually, what Jay had wanted me to talk about is to do an update on the new round of what's happening in the world of genetic engineering, or what I'd like to call GE 2.0, although it is the same old. So I'm going to talk about gene editing, gene drives, and some of their problems, and then also try to give folks a little bit of sense of hope. So first we have to ask, this is a short review, I'm sure most of you know it, but genetic engineering, the first crops that came on the market were in 1995. The major crops that are on the market right now, it's still basically these two traits, herbicide resistance. The vast majority of that is glyphosate tolerance, although because of increasing resistance, they're turning to 2,4-D and dicamba, which cause their own problems. And then there's insect resistance, the BT, which has also led to resistance as well. And then there's a very tiny bit that's virus tolerant. But the herbicide tolerant and BT are by far the vast majority. So if we look at the main crops and what's been engineered, soybeans, 94%. The herbicide resistant, this is all glyphosate. Sugar beets, 99%. Glyphosate, corn, which is both BT and herbicide resistance, and you can see multiple stacks. That's up at 93% for total acreage. 89% has their glyphosate tolerant trait in it. Canolas, 93% glyphosate. Cotton, which is both BT and herbicide resistance, 96%. 79% of that acreage has their glyphosate tolerant trait in it, or increasingly dicamba and that. Then the papaya, which is virus tolerant, is 80% in California. And then we don't have any updated data, but it's still probably around 13 to 15% for squash and zucchini, which are virus tolerant. If we then look, the vast majority of what this trait has done is basically increased use of glyphosate. Here's the most recent figures. That's, I believe, 2017 data, and they increased through time. And you can see the main crops: corn, soybeans, cotton, and alfalfa. Those are all engineered for herbicide resistance. Oh, I didn't include alfalfa. That's about 99% resistance. So this huge increase in glyphosate to make it the largest used insecticide in both the US and the world by far. And people have heard about all their problems. So what the answer that the biotech companies have come up with is, rather than diversify, they're talking about these new forms of genetic engineering, which of course, unlike the previous forms, are going to be highly specific. And these forms of gene editing, there are four major classes of what we call them. There's the meganucleases, the zinc finger nucleases, TALEN, which is transcription activator line E effector, and then this last one is the CRISPR-Cas system. And in all of them, what you're functionally doing is putting a guide like RNA in, and then you have a protein, which is like a molecular pair of scissors that cuts the DNA on both sides. And the idea is now you can precisely target things. If, for example, in hemophilia, you want to get rid of that, well, we just cut out the one nucleotide base mutation that causes that, replace it with the correct one. So they say this is incredibly exact. And even the words they're using, gene editing, they say it's like when you edit a book. You're going in and you're making these very precise cuts. And they argue that this is much more precise than regular genetic engineering, where you're literally shooting things into the genome. You have no control over where they're where they're inserting themselves. And I sort of laugh at this argument. Why? Because it's the exact same argument they used with genetic engineering 30 years ago, is they told us this is much more precise. We're only using one gene. We're only moving one gene or two, not tens of thousands. And some of us were saying, but you don't have any control over where you're putting them. And they told us it was more precise and more accurate. And this is the exact same thing that they're trying to tell us with editing. And even the terminology makes you think that way, right? Because you think of a book editor, which is actually something that's very precise. Let's look at the CRISPR-Cas9 system, since that's the one that gets the most attention. And what you see here is there's this guide RNA. That's where you're trying to figure out the sequence you want to target to cut, right? So that's the genetic material. Then you have this Cas9. That's a protein, right? And that's the scissors which snips the DNA. So this is how this whole system, you can now target things. So they're going in and saying, we can do everything. We can cure diseases, we can engineer animals, we can actually do gene drives, which I'll talk about. Gene drives are where you can literally take a gene and then it actually, since we all have two copies, one from the mother and one from the father, with a gene drive, you put that gene in and it changes the other one that's on the other chromosome, so it basically drives a trait through the entire population. So the problems with gene editing is they said it was very precise and they can show where to cut, but of course, wherever that sequence is found in the genome, it's going to cut, right? And it turns out that anything that looks similar, it will cut there. So you can have all sorts of unintended effects. And what I like to say is, if you're thinking about this as editing, think about an editor that says, okay, we have duplicated the word "the." So we're going to go in and we're going to cut out THE. Well, the way this thing works is wherever it sees those three letters together, TH and E, it deletes it from an entire book. That is not precise at all. And it turns out that it not only cuts there, but it cuts at sequences that are similar or might be not so similar. So as an example, just this last year, they were able to show that it introduced operations not only at the target sites, but at far-flung different arrangements. They said significant on-target mutagenesis, which means where you're inserting, because the repair mechanism isn't exact, you can induce mutations. So as it notes, on-target mutagenesis, such as large deletions and more complex genomic rearrangements at the targeted sites in mouse embryonic stem cells and in a human differentiated cell line. They said these damages in mitotically active cells caused by CRISPR-Cas9 editing may have pathogenic consequences. So showing that since these are complex rearrangements, the simple way they look for off-target effects is by scanning the genome. And they're pointing out you're not going to see these, right? So that's one example. Another example is this off-target effect causing immune dysregulation, where this unintended repair of on-target genomic cuts caused this pathogenic bystander mutation that led to disruption in the immune system. This was just published last month. Yes, and they can cause cancer. There have been 2 studies recently that showed when they edited an animal cell, what happened is part of the repair mechanism triggered this P53, right, as part of the repair. And the P53, actually, it says CRISPR-Cas9 genome editing induces a P53-mediated DNA damage response. The reason that's important is, what is P53? What else does that enzyme do? Well, that it can cause cancer, not just occasionally. For the P53 mutations that are responsible for nearly half of ovarian cancers, 43% of colorectal cancers, 38% of lung cancers, 1/3 of pancreatic, stomach, and liver cancers, and 1/4 of all breast cancers. Needless to say, this wasn't the only study. Second study that came out about a month later also from one of the companies, and what they do is they say, well, this is a problem, but there are ways that we can work around this by coming up with more specific editing systems. And they were also were using CRISPR to edit white blood cells with the idea they could cure AIDS, right? Because you would edit the white cells so that if the HIV virus tried to come in, it would be immediately cut up. And what happened is they had this idea, when they tested it within cell cultures, it took the HIV virus less than a week to evolve resistance. And in fact, it used some of the CRISPR stuff to become resistant itself. So this gene editing is not so good. The other thing they're now talking about is these gene drives, and that's where you edit in this gene, which then automatically corrects the corresponding gene on the other chromosome. So now all offspring will always carry that trait, and it drives through a population. So the big suggested uses they're talking about right now is we can cure malaria because we'll just eliminate the Anopheles mosquito, or the you can go after Aedes aegypti. So they're talking about it for disease stuff. They're talking about its use in Hawaii for introduced birds and other things. They're figure out ways or introduced mammals. They're thinking about doing it on just off of Martha's Vineyard on a small island where these rodents have come in. And part of the problem with the gene drive is once you put it out there, it'll drive through the whole population. If it escapes and goes into any other population, it'll drive through that as well. So there are real problems, and when you look at the logic of this, they've actually said this, that part of the way to deal with glyphosate-tolerant weeds is, well, you can just use these gene drives. So those weeds like the Palmer amaranth, you can just put a gene drive in them, and you'll make them susceptible to Roundup and dicamba and 2,4-D. So that means you can still continue to use this crazy pesticide-laden system, because what you're doing is the pests, the weeds and that have become resistant, well, you can just use a gene drive. This is the economic logic of this system. So let's look at this. The GE approach to control diseases is in mosquito control is this example from Oxitec. It's not quite a gene drive, but what they did is they engineer a gene that basically leads to lethality. And they're trying to control both the Asian tiger mosquito and then Aedes aegyptica. And the two ways to do it is there's this using this Oxitec, these genetically engineered mosquitoes, which basically lead to infertility. So you have to ask, well, then how can you raise them in the lab? And what they do is they put this little genetic promoter on, and in the presence of tetracycline, the gene is turned off. So that means the animal lives, right? So that means in the lab you can rear the males because you basically put tetracycline in the food. When you release them and they mate with the females, that trait is there, and unless there's tetracycline in the environment, then all the offspring are sterile. So it's this trait that you can drive the population. And Oxitec has done research down in the Cayman Islands and in Brazil and that. They've claimed they've gotten a lot of success, but the data is really, really iffy, and they found that there's been problems of up to 15 to 20% of the males end up surviving because tetracycline, at least in Brazil, can be in the environment because of its heavy use in agriculture, so it can be in dung, it can be in all sorts of things. Now, the alternative is here we have genetic engineers trying to figure out how things work in the, and how to engineer insects. And we have very little understanding. And yet there's actually a natural bacteria that was off in the weird part of the world, this Wolbachia. It's in about 60% of all invertebrates. And it was ignored for so long because it's just this strange little bacteria. Well, it turns out what it does is it can increase lifespan of insects, it can decrease them. There are some species which are all females because the Wolbachia has basically eliminated the males, so it can change sex, it can do all sorts of things. It's sort of like a natural biological control, because what you can do is you can take a mosquito, infect it with a Wolbachia. When it then mates with a female, when you release it in the wild, there's the cytoplasmic incompatibility, so all the eggs are sterile. So it's doing the same thing that the genetic engineering does, only it's evolved to do that. So you might want to ask, how successful have these things been? Well, the current status is for EPA, for this Wolbachia use, the company is called Mosquito Mate. It's a scientist from the University of Kentucky, and they have this with Wolbachia pipiens for Aedes albopictus for 5 years. For 20 states, now these males can be released. And also last year they did an experimental use permit so that these infected strains of Aedes aegyptica male mosquitoes are being tested in California and Texas and Florida. In Fresno, where they're being tested just this year, 95%, they saw 95% reduction. So this is working very well. The Oxitec still hasn't gotten off the ground. They haven't had their approval for any of the releases yet. And then the other thing that I want to talk about is, in this country, you have to take this mosquito mate control, right? Because you're still knocking down a population, which is good, but you might disrupt the ecology of the situation, because what happens when that species is no longer in there? But for this technology to work, you have to be able to basically to continue to sell something, because you have to continue to release these males. Well, another way you can use Wolbachias or other strains which cause this incompatibility. So what they do is they infect the male, and when they release that, it breeds with the female, and all it does is it doesn't affect the only thing it affects in the female, it makes them unable to transmit flaviviruses. So that means Aedes aegyptica, which transmit Zika and dengue and chikungunya, the mosquitoes still bite, they just can't transmit any disease. So that's a very effective way, because you're not really disrupting the ecology of the system. The mosquitoes are all still there, but they don't transmit disease. This is actually what's being used now in Australia and throughout Southeast Asia. This has to be done through an NGO or the World Mosquito Program. Why? Because that's a product that sort of destroys its own market. You release it and it spreads by itself. So there's something that can't be sold here, right? And that's exactly the kind of public sector research that why we set up the land grants in the first place, was they were supposed to be doing research that was for the benefit of the public, for the general public, not for the markets. Markets can do their own thing, and that's what, basically, that's what we have with all this pesticide market and all this focusing on GE, right? You're trying to develop products that you can sell. Whereas if you take a more holistic approach, here, like with this mosquito control, you learn from nature. You find these obscure bacteria and you learn how to work with them to get these nice controls, right? That's the kind of work that should be done in the public sector, not, increasingly what we're doing now is the public sector is serving to do all the basic research so that private companies don't have to do that, so they can wait and just get the product that they want to commercialize, when in fact these other things that have clear benefit to the public but the market can't provide, that's the kind of stuff that should be done. And that's also what organic and agroecological approaches are showing as well. So I just want to say that even though there is this focus on the bad forms of GE, there are people working in ways to use ecology and organic things to actually solve problems, and these aren't being paid attention to. People are always talking about the GE stuff, and it's not working. They're not talking about some of these other things that work, and part of the reason it's not discussed is because of this whole economic issue of, since it's not something that can make money, there doesn't seem to be a reason to push it. So thank you.
Okay, we have a break now.